FDA's office clearly stated in his summation that the tomatoes did not demonstrate a reasonable certainty of no harm. The FDA's Additives Evaluation Branch also was in agreement that unresolved questions still remained. The political appointee, however, did not require that the tomato be withdrawn from public consumption. According to Dr. Arpad, one of the world's leading experts in GM food safety assessments, the type of stomach lesions linked to the tomato could lead to life-threatening hemorrhage, particularly in the elderly who use aspirin to prevent blood clots. Dr. Putzi further went on to state that the digestive tract, which is the first and largest point of contact with foods, can reveal various reactions to toxins and should be the first consideration of GM foods risk assessment. Dr. Putzi was the first to sound the alarm that the limited studies conducted on the flavor-saver tomato never looked past the stomach to the intestines. Monsanto breezed past these findings and continued to market the flavor-saver tomato. Eventually, the flavor-saver tomato was taken off the market, but after this, no other biotech company has ever submitted detailed data to the FDA. Next, I'll discuss the main detoxifier for the body, and this is the liver. And the state of this organ is another indicator of the presence of toxins. In further independent studies, microscopic analysis of the livers of mice fed Roundup Ready soybeans revealed altered gene expression and structural damage. Rats fed GNA lecithin potatoes had atrophied livers. Rats fed Monsanto's MON 683 corn, which by the way was genetically engineered to produce Bt toxin, had liver lesions and other indications of toxicity. Rabbits fed GMO soy also had altered enzyme production in their liver and abnormally high metabolic activity. The livers of rats fed Roundup Ready canola were 20 to 25% heavier, possibly due to liver disease or inflammation of the pancreas. These studies are not random and, according to molecular geneticists, reflect damage on the liver by GM products. Studies over a period of the next 2 years reveal reproductive failures and infant mortality in both rats and mice fed Roundup Ready soybeans. In rats, the reproductive organs were dark blue instead of the normal pink color seen in healthy rats. In mice, young sperm cells were altered, and the embryos of GM soy-fed mice also showed changes in the DNA function. Even more dramatic results were discovered by leading scientists at the Russian National Academy of Sciences. Female rats were fed GM soy beginning 2 weeks before they were mated. Over a series of 3 experiments, 51.6% of the offspring from the GM-fed group died within the first 3 weeks, compared to 10% from the non-GMO soy group and 8.1% from non-soy controls. High pup mortality was characteristic of every litter from mothers fed to GM soy flour. The average size and weight of the GM-fed offspring was much smaller, and in later studies, the surviving GM-fed offspring were unable to conceive. Even more startling and bizarre, if that's the word appropriate for me to use, after the 3 feeding trials, the supply of rat food used at the Russian laboratory began using GM soy in their formulations. Since all the rats housed at the facility were now eating GM soy, no non-GM fed controls were available for further study or GMO feeding trials. All follow-up studies were then canceled. After 60 days on the GM soy diet, the infant mortality rate of all rats throughout the facility has skyrocketed to 55.3%. Without even trying, GM foods have produced a perfect means of keeping the rodent population down. Moving along, I'll discuss GM crops and the immune system and possible allergic reactions. Allergic reactions occur when the immune system interprets something as foreign, different, and offensive and reacts accordingly. All GM foods, by definition, have something foreign and different about them, and several studies reveal that they provoke reactions. Rats fed Monsanto GM corn, for example, had a significant increase in blood cells related to the immune system. GM potatoes caused the immune system of rats to respond more slowly, and GM peas provoked an inflammatory response in mice, suggesting that it might cause deadly reactions in people. It might be difficult to identify which GM foods were triggering allergic reactions in the population, since very few countries conduct regular studies or keep accurate records. One country that does have an annual evaluation is the U.K. After some GM soy was introduced into the British diet, researchers at the New York laboratory reported that allergies had skyrocketed as much as 50% in a single year, although no follow-up studies were conducted to see if GM soy was indeed the cause. There is evidence showing several ways in which it might have contributed to the rising incidence of allergies. The only significant variety of GM soy is Monsanto's Roundup Ready variety, planted now in 89% of U.S. soy acres. A foreign gene from bacteria, which parts of a virus and petunia DNA is inserted, which allows the plant to withstand the Roundup herbicide, also known as glyphosate. The protein produced by the bacterial gene has never been part of the human food supply. Because people aren't usually allergic to a food until they have eaten it several times, it would be difficult to know in advance if the protein was an allergen. Without a surefire method to identify allergenic GM crops, the World Health Organization and others recommend examining the properties of the protein to see if they share characteristics with known allergies. One method is to compare the amino acid sequence of the novel protein with the database of allergens. If there was a match, according to the World Health Organization, the GM crops should not be commercialized. Sections of the protein produced in GM soy are identical to shrimp and dust mite allergies, and I don't need to comment and tell you how many people are allergic to shellfish and dust mites. Fortunately for Monsanto, their GM soy was introduced before the World Health Organization criteria was established, and the recommended additional testing was never conducted. If the protein does trigger reactions, the danger is compounded by the findings that the Roundup Ready genes transfer into the DNA of human gut bacteria and may continuously produce the protein from within our own intestines. In addition to the herbicide-tolerant protein, GM soybeans contains a unique, unexpected protein, which likely came about from the changes incurred during the genetic engineering process, which I discussed in part 1 of my report. Scientists found that this new protein was able to bind with IgE antibodies, suggesting that this may provoke dangerous allergic reactions. The same study revealed that one human subject showed a skin prick immune system response to GMO soy only, but not to natural soy. Another study showed that the levels of one known allergen, trypsin inhibitor, was such as 7 times higher in cooked GMO soy compared to a non-GM control. GM soy also produces an unpredicted side effects in the pancreas of mice. The amount of digestive enzymes produced is dramatically reduced, and a shortage of enzymes caused by food proteins to break down more slowly, then they have more time to trigger allergic reactions. So what this could mean in digestive problems from GMO soy might denote allergies to a wide range of proteins and not just soy. The higher amount of Roundup herbicide residues on GM soy might create reactions in consumers. In fact, many of the symptoms identical in the U.K. soy allergy are among those related to glyphosate exposure. The allergy study identified irritable bowel syndrome, digestive problems, chronic fatigue, headaches, lethargy, and skin complaints, including acne, all related to soy consumption. Symptoms of glyphosate exposure include nausea, headache, skin rashes, and burning or itchy skin. It is also possible that glyphosates break down AMPHA, which accumulates in GM soys after they have been sprayed, and this might further contribute to allergies. It is interesting to note that in the 5 years after the introduction of GM soy, U.S. peanut allergies have tripled. It is a known fact that a protein in natural soybean cross-reacts with peanut allergies. Therefore, soy may trigger allergies in some people who are allergic to peanuts. Given the startling increase in peanut allergies, scientists should investigate whether there is indeed a cross-reactivity amplified by GM soy. For years, organic farmers and others in the commercial agricultural business have sprayed crops with solution containing natural Bt as a method of controlling insects. The Bt toxins create holes in their stomach and kills them. Genetic engineers take the gene that produces the toxin in bacteria and inserts it into the DNA of crops so that the plant does all the work and not the farmer. The fact that we consume this toxic pesticide in every bite of Bt corn is very unappetizing. Biotech companies are quick to point out that Bt toxin has a history of safe use and is rapidly destroyed in our stomach and won't react with humans. Independent studies, however, verify that natural Bt toxin is not fully destroyed during digestion, and it does react with mammals. Mice-fed Bt toxins, for example, clearly showed an immune response as patent as cholera toxin, and the mice had damaged and altered cells in their small intestines. When natural BT was sprayed in Washington state to fight the gypsy moths, over 500 people